my own, which is focused on the possibilities of chromatography for the separation of various types of isomers of lipids, which is based on more than 20 years of experiences of my group with LC-MS analysis of lipids. So really many people contributed to this work over the years. So I'm not able to list all of them in the title here. So the first question is, why chromatography? Do we really need this tool for the lipidomic analysis in case we have really ultra-high resolving power mass spectrometers like ICR or Orbitraps? So the answer is obviously yes, because the chromatographic separation can add the dimension, especially for some isobaric and isomeric species, how to resolve them, which may be difficult by MS alone. So if you look at this molecule of glycerol phospholipid, glycerol phosphocholine, so first we start with polar head group. Here you have a couple of examples, and you know much more from the Lipid MAPS classification, that there is quite many building blocks how to assemble various lipid species within lipid categories and lipid classes. So if you look here on aliphatic chains, so it may be either acyl, alkyl, alkenyl. The chain may have a different length. It may be linear or branched. It could be saturated or unsaturated, and it may be different number of double bonds, different positions, and of course, different geometry. And finally, there are different positions on the glycerol backbone. So we may differentiate regioisomers or enantiomers. And I will show you a couple of examples how you may deal with different type of lipid isomerism. You know well already this classification according to Lipid MAPS. So just to emphasize that in my lecture, I will deal only with 5 highlighted categories, not all of them. So this is the outline of my presentation. So after a very brief introduction, because Maria Fedorova already introduced well LC-MS, so then I will start with separation of lipid classes, then lipid species. Chapter 4 is the separation of various types of lipid isomers, and finally just some special application of 2-dimensional chromatography. So my introduction is really very brief. Just to remind you that the chromatographic process is basically the dynamic distribution of analytes between 2 phases. One is called stationary phase, and this is typically packed in a column. In case of liquid or supercritical fluid chromatography, there are other options with paper or TLC, but we will not deal this in my lecture. And the second phase is mobile phase, which is liquid in case of LC, or supercritical fluid for SFC, or it could be gas for gas chromatography, not covered in my lecture as well. And finally, the last step should be definitely mass spectrometry for detection, because for all omics techniques, including lipidomics, it's really needed, and the universal detection like UV detection will probably not be sufficient. Now I would like to start with the explanation of differences between lipid class and lipid species separation. We have prepared the artificial illustrative mixture of 4 molecules, 2 phosphatidylcholines with long chains and short chains, and of course corresponding lysophosphatidylcholines with the same chains. So if you look at lipid class separation, you have to realize what is the primary interaction here, and this is between the polar part of the molecule. So you see schematics here that we have choline and phosphate group. It means many hydroxyl groups there. So this interacts with polar part of stationary phase. This could be either hydroxyl groups on silica gel or water adsorbed on the surface in case of silica. So basically this is polar interactions. So it means more polar groups, stronger retention. And the effect of fatty acyl chains is very small to negligible. So you obtain individual classes separated in single peaks, but of course there is some in-class minor separation as well, will be shown later on. But if we do lipid species separation, typically with reverse phase chromatography, then the primary interaction is interaction between hydrophobic part of lipid molecule with non-polar part of stationary phase, for example, octadecyl silica gel. So simply longer fatty acid chain, higher retention is. But we have also separation within individual classes. So typically you are able to separate much more molecules, including some isomers. But this could be some difficulty for quantitative analysis. So this is the major difference between class and species separation in lipidomics. And here I would like to fairly summarize really key major advantages and drawbacks of both methods. And all these parameters summarized here are really important. So if you look for class separation, the key advantage is for quantitation, because you have the collusion of class internal standard with analytes, so the quantitation is much easier and more robust. There is also quite easy to do identification and automation of the whole process. But the drawback is that you have just summary formula. It means only the carbon numbers and double bond numbers, but you are losing the isomeric separation, which is, by the way, mirrored here, that this is the major advantage of lipid species separation by reverse phase chromatography, where you are typically able to detect about 2 to 3 times more species, but you are limited concerning the quantitation. I don't claim that quantitation is impossible, but definitely you need to put much more emphasis on the use of multiple internal standards per class and to do proper analytical validation to be sure that your quantitation is not influenced by different matrix effects for internal standards and for analytes. And ID is also more laborious. So first, I'm starting with the explanation of these 3 lipid class separation methods. So the first one is called normal phase chromatography. This is something quite historical, and I guess not many of you can use this. Stationary phase here is more polar than mobile phase, and the typical stationary phase is silica gel or aluminum oxide or some medium polar bonded phases. Mobile phase is totally non-polar, hexane, heptane, with a very small additive, very small percentage of polar modifier. And this can be applied well for non-polar lipids like glycerolipids, cholesterol esters, and so on. It's compatible with APCI, APPI, but not with electrospray. And here we also have lower reproducibility because of huge sensitivity to traces of water. So definitely, I don't encourage you to use this method if you are really not experienced chromatographers, because it's not so easy to handle. Just one example that you may separate various acylglycerols, cholesterol esters using this methodology and conditions given here. At present, I would say that normal phase chromatography was almost completely replaced by hydrophilic interaction chromatography, abbreviated as HILIC, which is definitely more user-friendly, and I can recommend to use it because it's very simple and convenient for quantitation. Here, we have polar stationary phase and polar mobile phase, which explains why non-polar lipids cannot be retained in this system and non-polar lipid classes are eluting in the void volume of the system, such as triacylglycerols, cholesterol esters, and so on. And you cannot improve it and separate them due to the characteristic of this system. Typical stationary phases are silica gel or some modified medium polar stationary phases, and mobile phase acetonitrile water, methanol water, always with high content of water and also additives. Application almost to all lipid classes except for non-polar lipid classes. Here we have relatively low back pressure of the system. It means that we can use higher flow rate and shorten the analysis. And here you see that you have also partial separation inside individual classes. So this is the example of HILIC separation of the mixture of standards of various classes of phospholipids and sphingolipids. So you can see that within like 10 minutes, You may separate many polar lipid classes, but unfortunately, non-polar lipid classes and some ionic lipids are coeluting in the void volume of the system. It means that you are able to quantify also cholesterol esters and triacylglycerols, but not di- and monoacylglycerols due to common fragment ions with PGs. Now I would like to show you some smart applications of HILIC applied for various classes of glycosphingolipids, both neutral and anionic. So you know already this nomenclature from previous lectures. So we have separated many neutral glycosphingolipid classes in the first paper, and then in the second one we have focused on gangliosides. So the first example is shown here for Cerebrosides and Globosides analyzed in human plasma. So the red record is positive ion mode and blue one is negative ion mode. So you see a couple of Cerebroside classes and also some other sphingolipids and very polar phospholipids separated here. Of course, if you use the convenient internal standards, they may be quantified as well. Here, I would like to show you a very smart way of identification of these glycolipids, because when you perform MS-MS, it's quite comparable picture as for example, proteomics or glycoproteomics, because you have convenient fragments to determine the positions of individual sugar units and even the branching. So this is the example of Globosides for in this case. So all fragment ions can be easily assigned and identified. The second example is anionic glycosphingolipids. It means that we should use in this case negative ion mode. And here I'm presenting the separation of 19 classes of gangliosides, ganglioside subclasses with some other sphingo as phospholipids separated as well. This is the analysis of porcine brain extract. And the conditions, silica gel column and the gradient of acetonitrile, aqueous ammonium acetate, and added acetic acid as well. And again, here, all these units may be easily identified. I mean, the sequence of saccharide units and the branching may be easily detected based on MS-MS experiments. So here is just one example of MS-MS identification of GT3 acylated. So you see that step by step, you observe all important fragment ions. So if you are interested in more details, definitely you can go into the paper and check it. Now, I would like to continue with the supercritical fluid chromatography. This may be something relatively novel for many of you, or especially the application in lipidomic analysis, but it's pretty natural to use this technique for lipids because it's well convenient for molecules containing large hydrophobic part. In this lecture, I'm not going into details because I have separate lecture for this topic. So I just show you this phase diagram from school textbook. So you see that we should operate here in the supercritical region or eventually in this subcritical region here. It means above critical point. It means this critical temperature and critical pressure. So almost exclusively carbon dioxide is used under these supercritical conditions. So the methodology will be explained later, but I will show you some results. So here you see our first paper on ultra high performance supercritical fluid chromatography MS analysis of both nonpolar and polar lipid classes. And you see that within less than 6 minutes, we are able to separate 30 lipid classes or more. The important issue is that there is a very nice separation also for nonpolar lipid classes, and this is why we favor this method over HILIC, because in case of HILIC, all these classes will be coeluting in the void volume of the system. So we can do both. And conditions are published here, super or later on subcritical carbon dioxide with modifier methanol, small amount of water, and also ionic additive. Here I would like to illustrate that inside individual lipid classes, we have also minor secondary in-class separation. So here you see reconstructed ion currents for triacylglycerols with different number of double bonds. So you see that there is a very small shift. You see here in minutes that the shift is extremely small. So in reality, you see them just within one peak. But if you reconstruct individual ion chromatograms, you see the partial separation and the logical retention order as additional confirmation of your identification. In case of carbon number, you see also logical order here for nonpolar lipids and vice versa for polar lipids, eluting at later retention times under different chromatographic mechanism. But I'm not going into details of mechanism here. Now I would like to present the comparison of results obtained by UHPLC-MS here with UHPSFC-MS. So the major difference is that in case of LC, we are not able to resolve non-polar lipid classes, which are well separated here, and SFC is also a little bit faster. Here it's about 4.2 minutes compared to LC 6.5. And honestly, with SFC, we have analyzed several thousands of samples already, maybe close to 10,000 samples were analyzed by this technique. And here you see the comparison of quantitative data from the same sample measured by both methods. This was already presented in this paper, so you may go into details if you like. And finally, I would like to highlight on these two slides benefits of class separation for quantitation. So if you just consider general bioanalysis and compare with specific features of lipidomics, so in both cases, we should start with the addition of internal standard before any sample handling to the sample. It means before the extraction. Then here, we typically use internal standard for each compound to be quantified. This is the standard in pharmaceutical industry, for example. And the best internal standard is isotopically labeled analog. This is the perfect way. In case of lipidomics, we claim that at least, at minimum, one internal standard for each class should be used. And this should be, of course, some exogenous or isotopically labeled lipid class internal standard. The next step is the same, that internal standard and analyte should be ionized at the same time to avoid different matrix effects. And this is really a very important issue which should not be underestimated. So this is perfectly fulfilled here because we have standard for each. But in case of lipidomics, this requirement is fulfilled just for lipid class separation or shotgun approaches, but not for reverse phase chromatography. So the care really should be taken when you do quantitation with reverse phase. Here you see the peak of phosphatidylcholines in case of HILIC analysis, and in different colors you see various internal standards used for quantitation of this class, and the corresponding mass spectrum is shown here. So we really prefer to use multiple internal standards per class, because then you may use one internal standard for quantitation of the second one, because you know concentrations of both of them. So this way you are able to calculate the quantitative error of your determination, and then you will be definitely much more confident with your quantitative data. And you may also avoid some possible mass interference if you use just low resolution analyzer, or if you have some hidden problem. This way you will easily discover this problem. And this is just the list of internal standards, what we are using in our laboratory for lipidomic quantitation of these 5 lipid categories. I'm not going into the details. Basically, we have deuterated, shorter chains, longer chains, and in some cases, odd carbon fatty acyls. The key issue is to verify that these lipids are not occurring in your real biological samples. Then you can use them. But of course, isotopically labeled are the best, but also the most expensive. Now, I would like to explain the benefits of species separation by reverse phase chromatography. Here we have non-polar stationary phase and polar mobile phase. Stationary phase, like long chain bonded to silica gel, like octadecyl silica gel, octyl, or others, eventually polymeric stationary phases. And mobile phases are typically methanol water, acetonitrile water, maybe some additives, or in some specific cases, like for triacylglycerols, you may skip the water, and then we call it like non-aqueous reverse phase chromatography. Applications are almost universal except for ionic lipids, which may elute close to the dead volume of the system. A very useful parameter here is equivalent carbon number for calculation of retention behavior, which is calculated by the total carbon number in all fatty acid chains minus two times of the number of double bonds. And then you can predict retention windows for individual species. This is by far the most widespread methodology in LC. And higher polarity means lower retention. Now I am going to back to history. This is my first ever paper in the lipid analysis, published already in 1999. The analysis of the reaction mixture during the biodiesel production by reverse phase chromatography, non-aqueous reverse phase chromatography. So if you see the big widths, it's quite funny from the current perspective. But we were able to separate all these tri, di, and monoacylglycerols, fatty acyls, and also final biodiesel product, methyl esters. A few years later, we continued further, and we separate many isobaric tri and diacylglycerols according to these ECN values. But if you see the analysis time, almost 2 hours. This is something unbelievable from our current perspective, because nowadays we can do the same within like 20-25 minutes. Then we continued with the quantitative aspects that we have realized that not all responses are identical. So we have introduced the use of response factors, used also by Gerhard Liebisch and other groups, to improve the quantitation and to consider that the response may depend on the acyl chain length and number of double bonds. So the final summary of this is shown here in this paper, that in total we have unambiguously detected 264 triacylglycerols containing 28 fatty acyls with 6 to 26 carbon atoms and up to 4 double bonds. In all cases, we are of course absolutely confident with identification, because we use various tools to be really sure, and we never present tentative identifications or just some features. And I will explain now. I return back to the current days, and I would like to present here the reverse phase UHPLC methodology, what we are using at the moment for multiple lipid classes. So this is the example of separation of human plasma in positive ion mode, and you see many classes, and the retention windows of individual classes are heavily overlapping. So the identification is a little bit tedious and really not trivial. Some lipid classes are better ionized in negative ion mode, as reasons explained by Maria Fedorova already. So you see that some classes are better expressed here. And concerning the confidence of identification, I would definitely emphasize the role of regular retention behavior of lipids. It may be used for both lipid class and lipid species separation methods, but in case of reverse phase chromatography, these dependencies are the best, I would say. So for example, here, if you select some lipid class like diacylglycerols, you take the constant number of carbon atoms and different number of double bonds, you may put it in very nice dependence. And you may do the same for all lipid classes. So we are able to create over 50 various dependencies, and they are in all cases better than 0.999. Here you may see multiple species. It means that you have different isomers of double bonds. So if you select only one logical series, then the fit will be perfect, like in this case. I will show it later. The same dependencies may be created also for the situation where you have constant double bond number and varying fatty acyl chain length, as shown here. So if you properly define these logical series, then these dependencies are really perfect. By the way, if you look for some missing members in this logical series, it's very easy to very accurately predict the retention time and then search in your record whether this species is present or not. So if you see these correlation coefficients, it's quite accurate. And vice versa, you may also use it to detect some suspicious species or incorrect identifications. So that's why I'm going towards this approach for this highest possible confidence of identification, what I like to call 0% false rate discovery, that before you present any lipid detected in your biological system, you should do everything possible to exclude possible mistake. So we propose here to verify by the logical retention behavior, at least by the two dependencies, as shown here for lysophosphatidyl-cholines. You see that even regioisomers are resolved in this curve. So in one case, constant fatty acyl chain length. In second case, constant double bond number. And then we are really confident with the retention times. Then, of course, all mass accuracies for multiple ions in MS first, MS second, and both for positive and negative should fulfill the criteria of given mass analyzer. In our case, for example, better than 5 ppm. And if you do this, then you are really highly confident about your identification and you should not over-report. A few illustrations to show the really power of reverse phase chromatography and the added value in case of separation of isomers. So if you look here on this group of phosphatidylcholines, so if you have 0 double bond, just single compound, the same for mono unsaturated, but when you have 2 or 3 double bonds, then you see different isomers differing in the position of double bond and or different distribution of carbons in fatty acyl chains. So the same may be observed here for sphingomyelins, but this is even more complex. Reverse phase chromatography is the, I would say, the only suitable technique for the analysis of oxylipins. The reason is quite similar, quite simple, that there are so many isomers and different positions of hydroxyl groups and so on, that you really need the best possible separation mode. Otherwise, you will mix up them together, and you don't like this. Definitely for these molecules, negative ion mode is better. So I show here this separation of 63 standards in 12 minutes together with deuterated internal standards. And this was applied then for analysis of oxylipins in human plasma. But in the literature, you may find even better examples to find more species. And this was already presented also here by Ed Dennis, that even more oxylipins could be detected in biological systems. So now I would like to show you the separation of various types of lipid isomerism step by step. So it means the different length of individual acyls, distribution and positions of double bonds, geometry, and also regioisomers, and finally enantiomers. So I start with this example of different positions of double bonds. It's well known that for conifer seed oils, for example, large shown here, there are some unusual double bond positions. For common fatty acyls, you have the positions 9, 12, and 15. But in case of these conifer seeds, the first double bond is in the position 5. And then you may observe here that we always have a logical increment in retention, calculated here as a retention factor, which contributes to confident identification of individual unusual fatty acyls present either 1, 2, or 3 times in particular triacylglycerols. Further, you should check also mass spectra, and you see that the ratio of protonated molecule and this first fragment ion after neutral loss of fatty acid is quite characteristic for these unusual double bond positions. Of course, we have also verified by available internal standards that we are really correct. Then the second type of isomerism is different geometry of double bond, which may be separated either by silver ion chromatography or very long reverse phase chromatography with multiple columns. So silver ion chromatography, this is something really very rarely used, but in past it was quite common in lipidomic analysis and using TLC chromatography, for example. This is based on the interaction of silver ion with double bonds, with reversible interactions. And simple rule is more double bonds, stronger interaction, higher retention. You need the dedicated silver ion columns based on cation exchanger. They are pretty stable, no leak of silver into mass spec. And with optimized system, you are able to resolve cis-trans isomers, regioisomers, and also double bond positional isomers. So first example is shown here for triacylglycerols that you see that the retention of all trans isomers is much lower compared to cis, and it's relatively close to saturated species in line with known space configuration of trans double bond, which is much closer to saturated chains than in case of cis configuration. And you see the same if you have 2 or 3 double bonds. Then again, for one trans, the retention behavior is retention times are significantly lower and could be easily differentiated. Another situation what can be resolved by reverse phase chromatography is here again we have cis-trans differentiation, but also the resolution of linear and branched fatty acyl chains. This was also confirmed by available standards, and this was for animal fat from fallow deer. So this is the possibility to separate even such isomerism, but the separation is pretty long, and you see that we have the coupling of columns in the total length of 45 centimeters, which is really very long separation. So different positions of on glycerol skeleton, it means regioisomers, again either silver ion chromatography or very long reverse phase columns. So here you see the randomization mixture obtained from tripalmitin, triolein, and trilinolenine, and this way you obtain 6 regioisomeric doublets and 1 regioisomeric triplet. So here you see that you are able almost baseline to separate all of them using silver ion chromatography. If you look on mass spectra of regioisomers, they are also different. So if you compare the ratio of these fragment ions, and if you are able to calibrate this using the standards, you may use it even for relative quantitation with a relatively good accuracy. I would call it semi-quantitation. And this is the application of this methodology for the determination of SN2 occupation in various plant oils and animal fats. So you see here that in plant oils, there is always strong preference for unsaturated fatty acyls, especially linoleic. In case of animal fats, there is the preference of saturated in case of pork and wild boar, but for other animals, it's something in between. So for such detailed studies, this methodology is available. And finally, the most difficult example, the most difficult isomerism is enantiomerism. So for this, you definitely need some chiral environment, either chiral selector bonded in the stationary phase, which is the approach what we have used, or chiral mobile phase, which would be terribly expensive and difficult to use, or chiral derivatization to obtain diastero isomers, which may be separated by conventional systems. So I will show just examples using these polysaccharide chiral selectors applied for acylglycerols. So there are no available standards on the market. So we have developed the procedure for their synthesis, first with the blockage of 2 positions, and then you implement just 1 acyl, and then after the hydrolysis, you introduce additional 2. And when you synthesize these standards, you may optimize your method, again, 1/2 meter chromatography column, chiral column, not cheap, definitely, but you are able to separate it. There is also possibility to use this randomization reaction for 2 monoacyl triacylglycerols, schematically shown here, and you obtain this mixture. And here you have 2 pairs of enantiomers and additionally also regioisomers. So when we look on the separation, again, this is normal phase systems. Normal phase systems, so more double bonds means stronger retention, but we are also able to separate enantiomers, red arrows, and regioisomers. So this is the example of possibility of chiral LC-MS. By the way, the mass spectra of enantiomers are identical. This is within the experimental error, and for regioisomers, there is difference. The same approach may be applied also for diacylglycerols and monoacylglycerols, and they can be also separated using this system. So finally, I'm going to the end of my presentation, 2-dimensional coupling of 2 different LC systems with different selectivity. So typically we combine lipid class separation and lipid species separation, but this is definitely technically demanding. There are 2 options, offline and online. I will show examples of both of them. So first one is offline separation of lipid classes by HILIC chromatography. This is the sample of porcine brain extract. And then you transfer this fraction to the second system, and you may resolve them, for example, by reverse phase chromatography. This is quite old work, so the resolution is not as good as we have at the moment. This is just for illustration. And here you see quite good coverage of separation space. So the peak capacity is very high, and the orthogonality of both systems is relatively good. Here you see that such very small differences may be resolved also by reverse phase chromatography. Here the only difference between oleic and allydic acids is the configuration of 1 double bond. But when we prepare the randomization mixture, we are able to separate them. But when we switch into this system to silver-end chromatography, we have also partial separation of regioisomers. So such fine details may be resolved only by chromatography. With MS, you may have serious difficulties. And the final chromatogram, what I'm going to show here, is the comprehensive coupling of reverse phase chromatography with HILIC online. So you see the coverage of separation space here. But of course, this is quite technically demanding. So you need some experiences with this methodology as well. So finally, I'm going to conclude my presentation. Just to summarize that I have tried to explain you what means lipid class separation and what is the advantage for the quantitation and automation of your workflows, but also what are the limitations and what is some complementary advantages of lipid species separation with better selectivity and more identified species. So in fact, if you imagine that you combine the benefits of lipid species separation and lipid class separation, this would be definitely the holy grail for lipidomic quantitation, but this is not available yet on the market, but we may see in the future. And we have also some special chromatographic modes for dedicated types of isomerism with tailored separations. So finally, I would like to acknowledge the funding and the contributions of my many present and past co-workers who contributed to individual papers and also prepared for me slides. And I would like to thank you for your attention. And at this moment, there is time for questions.